Hi, I'm Gerardo Ferbeyre. I'm a professor of biochemistry at the University of Montreal.
And I'm Emmanuelle Saint-Germain, PhD student in Gerardo's lab who conducted this research.
The p53 SOCS1 story is a project that's been going on in the lab for almost 10 years. And we published a paper in Molecular Cell 2009 identifying that SOCS1 can interact with p53.
So one of the reasons we chose to do this research was to try and better understand how SOCS1 regulates p53. So we already knew that p53 and SOCS1 interacted together and that this interaction was important in the induction of senescence by the oncogene STAT5. But we wanted to go more deeply into understanding how SOCS1 regulates p53 and what targets of p53 precisely does it regulate.
So the area of identifying p53 target genes is one of the most challenging areas in modern cancer research. p53 is the most famous and important tumor suppressor. There are thousands of studies on p53, and many of them try to identify which are the targets of p53 that explain why it's a tumor suppressor. Unfortunately, most of these studies don't overlap. It seems that p53 target gene regulation is very context dependent. So we took up to the challenge to identify in our own context, which is the senescence response to an aberrant cytokine signaling, in this particular context, what are the p53 target genes that we could identify?
So then what we thought was rewarding about this paper, we did a microarray analysis at first in a model comparing STAT5 senescence in which SOCS1 is overexpressed and STAT5 senescence in which we knock down SOCS1. So the cells with knockdown SOCS1 will bypass senescence, and we used these two models to try and identify the target genes that are dependent on SOCS1. And among those targets, p53 targets were found to be a major pathway, and we found many genes, among which we found SLC7A11, which was already shown by another paper to be a p53 target and an inducer of ferroptosis. So when we saw that, we were excited and we tried to see whether SOCS1 could actually have a direct impact on ferroptosis. And as a matter of fact, at the first trial, SOCS1 sensitized the cells really clearly to ferroptosis. So that was a pretty nice discovery to link SOCS1 and ferroptosis.
So the paper reports a really interesting series of p53 target genes, and I think it will stimulate further research into confirming that these are really p53 target genes using more genomic techniques like ChIP-seq and p53 targets that actually were not known before are suggested in our paper according to our genomic analysis. So I think that's something that we are very also proud of the accomplishment in this paper.
So a pathway that I would like to further explore in the future is the lipids, since we found a link between SOCS1 and ferroptosis, and in ferroptosis, oxidized lipids play a major role. We're really curious to see if overexpressing SOCS1 or down-regulating SOCS1 could have an impact on different oxidized lipids, and to characterize that a bit more.
Another area that we and hopefully other colleagues will explore in the future, coming from our paper, is the possibility of eliminating or killing senescent cells using the pathway we just discovered. It means that we could activate SOCS1 ferroptosis pathway in senescent cells to eliminate them. And this is a very hot area now in aging research. And there are wonderful demonstrations from several groups that show that if you eliminate senescent cells, you can actually increase health span in mice, and you can actually improve several age-linked disease. So these have been mainly done by using classic apoptosis in senescent cells, and I think our paper will stimulate to explore to induce ferroptosis in senescent cells.
Especially, we saw that one type of senescence was very resistant to ferroptosis induction, so it would be interesting to study that and to find what blocks ferroptosis in those cells, why they're resistant.
Finally, we thank AJ for a very efficient handling of our manuscript. Some of the reviewer comments were really, really useful and actually helped us to improve a lot our initial submission. So we're very happy with the outcome of this manuscript.